Hi everybody, welcome to the practical. This practical will give you a very quick introduction to very simple cell anatomy. We're going to use onions today for our cell anatomy explorations. Everybody knows what an onion's like, just the one out of the supermarket. We're going to use organic onions today. Organic onions haven't been sprayed with an anti-sprouting chemical. Sometimes if you have organic onions in your fridge, you'll find that they tend to sprout a little bit sooner than the conventional onions, which are usually in an orange net. Other items we need for today's practical: the tweezers. We're going to need a very sharp blade, just this one here, and we're going to use a range of stains as well. So the first thing to do is to get to grips with the anatomy of an onion. You probably haven't thought about this before. So if we were to look at this onion, you can see here are the roots. The roots would have been growing down into the soil. Here we have just basically a food storage area. So the bulk of the onion is specialized for food storage. And these are the withered, senesced onion leaves. So let's cut this down the middle. Now what an onion is actually a modified stem. Each one of these onion layers, is a modified stem which is specialized to store food. So it contains cells that are specialized to store food. So here we go. The part of the onion we want to explore today is the epidermal layer. Each one of these scale leaves has an epidermal layer separating it from the scale leaf next to it. So we need to go and search for the epidermal layer. The easiest way to do that is to count around about 3 scales in. So 1, 2, 3. Pull scale 3 and 4 apart. And what you're looking for is an almost slippy layer. Sometimes when you're cooking with an onion and you peel an onion and you're left with a slippy layer on the outside of the onion, that's the epidermal layer. So we need to find that. Now this side is quite smooth, so our layer must be in here on this side, which is good news because it makes it much easier to dissect. This is a good time to talk about the function of the epidermis of an onion. Here we have a rather moldy onion that I found, and I'm going to cut down through it and I'll talk you through what I'd like you to see. So obviously this onion has scales, and in between each one of the scales is an epidermis. And of course those scales, those brown papery scales on the outside, they're just old scales, and they're the first line of defense against fungal or viral infection. This onion has a particularly bad fungal infection, but you can see as I peel away these outer scale leaves that the inner scale leaves have escaped any damage. And that is because of that one single celled layer of epidermis. So to take a very small sample, what we're going to do is we're going to use a blade to cut a grid on the inside of this scale here. I'll just use a pen to show you what I mean. And what you're going to do is draw an X's and O's grid, like this here, on the inside of the onion scale. I'm going to do this again. I'm just using the pen to show you, but I'm going to do it again with a blade. And then all I need to do is pick off one of these small little squares, and that will give me my sample for the microscope. It's important to take the smallest sample possible, because if you don't, what happens is this epidermal layer naturally wants to fold over on itself. The beauty of using this epidermal layer is that it's 1 cell thick, and so we want to keep it 1 cell thick on our slide. I'm now going to cut my grid. Now I have my tweezers. It's a point-ended tweezers, which is important because I don't want to touch this epidermal layer too much. I might damage it. Then I've got my perfect little square of epidermal layer there. So just need a little bit of water, and you take your square and you put a few of those squares into the water, and that'll make sure that they're really nicely hydrated. It's always best to visualize any kind of a plant cell when it's fully hydrated. Here I have my epidermal samples in water. What I'm going to do now is to stain my epidermal peels. I'm going to use two stains. I'm going to use iodine, and I'm also going to use toluene blue. So as you can see here, the iodine is a brown colour. Iodine stains starch black or purple. So if that brown colour changes in colour to a black or a purple, that is a positive stain for starch. If it stays a brown colour, then it means that there is no or a very low level of starch in the sample. The second stain we're going to use for this experiment is a stain called toluene blue. Toluene blue will also allow us to diagnose the presence of certain molecules. For instance, in very thin sections, toluene blue will stain cellulose in cell walls pink. And in later practicals, you'll see where toluene blue stains lignin green. What we're ready to do now is move some of our onion sections into our iodine and move another few into our toluene blue. So they're moving them out of the water into the toluene blue. There we go. Just clean your tweezers in between so we don't get any toluene blue in our iodine stain. So we usually give them a little shake to make sure both sides of the section is covered in the stain. You could also use a dropper here. Just drop some more stain down on top. And we leave them then until they're fully stained and ready to be visualized under the microscope. I also want to set up another solution today, and it's a 1 molar sucrose solution, which is it's like table sugar. It's a highly concentrated solution, so when we place our epidermal peels, or our little squares, into this highly concentrated sugar solution, all of the water will rush out of the vacuole, and so the contents of the cell will shrink. That shrinking is called plasmolysis. And once you induce plasmolysis, you can see parts of the cell that you wouldn't usually be able to see when a cell is fully hydrated. So in this situation, the sucrose has been used as a treatment rather than a stain. To establish a 1 molar solution of sucrose, I first need to find out what the molecular weight of sucrose is. I can do this by adding up the weight of each one of the individual atoms of sucrose, or you can indeed just Google it. So when you Google it, you will find that the molecular weight of sucrose is 342 grams of sugar per litre. I need a much smaller volume than 1 litre, so I'm just going to make up 100 mils of 1 molar sucrose solution. That is simply done by measuring out 34 grams of sucrose and adding 100 mils of boiled water. Then all you need to do is just to swirl the solution to make sure all of the sugar granules dissolve, and then let the solution cool down before you put in the epidermal pieces. Once our sugar solution is cooled, we'll transfer a small portion into a smaller container. You can see here that this sugar solution is highly concentrated. It appears really syrupy, and of course that concentration of the sucrose is now much higher than the internal concentration of the epidermal cell. So as I'm moving these epidermal pieces into the sucrose solution, the vacuole, which is the major reservoir of water in the cell, is starting to empty, and what's driving that is the high concentration of sucrose on the outside of the cell and the low concentration of solutes on the inside of the cell. And this is osmosis in action. You've got a cell. Each one of the cells has got a semi-permeable membrane. There is now a concentration gradient. where the concentration of solutes outside the cell in the sucrose is very high compared to the low concentration inside the cell. And so water will leave the vacuole, go out across the semi-permeable membrane and leave the cell. During osmosis, it's always water that moves, and the direction of movement is always into the area that has the higher concentration of solutes. We are ready now to set up our onion epidermal slides. So a nice clean dropper, put a little bit of water on each slide, not too much. When you put too much on, it can spill underneath the slide, and that will cause your slide to stick to the stage of your microscope. It's just a case of finding a good section then. I have my slides labelled, as you can see here. It's very difficult to make a slide without bubbles, but one of the tips I have is that if you put your cover slip on at an angle so that the bottom of the cover slip catches the water, then if you just drop that cover slip, it should push out the air in front of the cover slip while it's falling. And that way you are more likely to get a slide that doesn't have too many bubbles on it. Whatever the case, it's good to know what a bubble looks like under the microscope. So here we have our nice little square section. It's not folded over or anything. And that will make looking at this under the microscope a lot easier. Our epidermal squares have been in 1 molar sucrose for 30 minutes. So they're now ready to be mounted onto a slide. The stained epidermal squares were mounted in water. However, we can't use water for our sucrose treated cells. For our sucrose treated cells, I need to mount them in a drop of sucrose. All this entails is putting a drop of sucrose onto the slide and then dropping a few of the epidermal squares into the sucrose. The reason they must be mounted in sucrose is because if they were mounted in water, osmosis would occur again, but this time the water would rush back over the cell membrane and would fill the vacuole, and therefore we wouldn't see plasmolysed cells. Instead, we'd see normal turgid cells where the vacuole is full and it's pushing all of the cell contents against the cell wall. For this particular experiment, we really want to empty that vacuole so that we can see parts of the cell, like the cytoskeleton. Before we look at your slide, we're going to have a quick introduction to the types of microscopes you would have used in WIT. So these are Olympus microscopes, and they're particularly good. The most obvious part of the microscope to point out first is the eyepiece, which is up here at the top, and they can move apart and up and down because everybody's face is slightly different in size, so you move those. So you look down each of these eyepieces, and this eyepiece has a 10x lens in there. So immediately you're getting a 10 times magnification just by looking down through the eyepiece. This here is the part of the microscope which actually allow you to fold the thing away. That's for storing. So this here is the stage. This is where you put your slide on, and you can see here that you have a slide clip. These here are objective lenses. They're all slightly different colour, slightly different size, as you can see there. So you'd always start with this 4x lens lined up. You start with a 4x lens. This one here is a 10x, you'd move up to this one then. And then you'd move up to the 40x lens. And then up to 100x. For plant biology, we'd never use this lens here. This lens is just for looking at bacteria. So if you have the 4x lens lined up, you're getting a 40x magnification. So you're magnifying your sample 40 times. The reason for that is this is 4x here. It's 10x up here. 10 times 4 is 40. So if you're using this blue banded objective lens, you'd actually be magnifying your sample 400x. So we talked about the slide. This is a stage where the slide goes on. It's got a clip in here. So this is the way you would use that. So the corner of your slide fits neatly in here, and your clip holds it in. And that's really important because this tool down here allows you to move the slide around the stage. So you can just go forward and back and from side to side. So that's called the stage directioner. Stage directioner. Probably the most important part of microscopy is making sure that the light is correct. So I'll just take this slide off so I can show you properly. There's obviously a light source down here, and there's a dimmer switch on it. You can turn it up and reduce the light. But underneath here, this item here is called a stage diaphragm, or the iris diaphragm. And you can see that there's a small handle here that allows you to turn up and down the light. So that's the iris diaphragm. If you look up the top there, you'll see when I move this handle that the light increases and decreases again. So that's a very important tool to use. When you use higher magnifications like 40x, the aperture, which is a little hole at the bottom of the lens, is quite small, and so you usually need to turn the light up to see your samples clearly when you're using the larger magnifications. The final items of the microscope then are the coarse focus and the fine focus. So the coarse focus moves the stage up and down, as you can see here, and only a little twist will move that stage a considerable amount. And as you move the stage, the plane of focus is moving. So if your sample is in focus, usually there's very little distance between your slide and your objective lens. So that's the coarse focus, it's this big wheel down here. Inside the coarse focus is the fine focus, it's this one here. And when I twist the fine focus, you can see that the stage is moving, but only very slightly. So the fine focus is used to get your sample into fine focus, to really sharpen your sample, because it only moves the stage a tiny amount, compared to the coarse focus, which moves it a lot. Let's visualize our onion epidermis stained with toluene blue using the microscope. Here you can see what I see when I use the 4x lens. That's the smallest lens. First of all, the thing to point out is the big bubble on this slide. The arrow shows where the bubble is. It's quite a large bubble, and it's quite difficult to get plant samples on slides without bubbles on them. But it's no big deal, because in this area in here, you can see there's almost like a brick effect, like a brick wall. And if you can use your imagination for a second, those bricks are the cells. So this brick wall is made up of many cells. You'll notice also that our toluene blue colour has now changed to a pink colour. And when toluene blue changes colour from blue to pink, this is a positive stain for cellulose. You'll see that it's stained at different intensities. So there's areas of this particular photo where it's hardly stained at all. There's a tinge of pink. And on the other hand, there are areas which are quite strongly stained and they're quite pink. These areas are strongly stained and are very pink because when I was taking the epidermal peel off the onion, some of the tissue from the scale leaf underneath the epidermis was still connected to the epidermis. So that strong pink staining is actually just some scale leaf tissue that is still connected on to those epidermal cells. The reason the epidermal cells don't stain very strongly is because they're actually quite simple cells, which is why we use them for this particular practical. When I say these cells are simple, it just means that they're not very complex. You know, they have a very thin primary cell wall, they'll have a nucleus, and then the rest of the cell is mostly just vacuole. And we all know at this stage that the vacuole is full of water and a few solutes. So they're not particularly complex cells. We've just been visualizing these cells at 40x magnification. What we're going to do now is zoom in and use that yellow banded objective lens, which is a 10x lens. So it means now our next visual will be our cells magnified to 100x. And you can see here in the screen where I've used the fine focus wheel to really get a nice crisp image. So you can see this brick effect here much clearly. And as I use the stage directioner to travel around the slide, you can see how each cell is connected to the one next to it. They're connected via the cell lumen in between the cell walls. And sometimes students think that the edge of these cells are quite sharp, but if you look really closely, you see that the edges of the cells tend to be quite rounded. That's an important detail to record in your drawings. You'll also notice that a lot of these cells don't seem to have a nucleus in them. Some cells have got a nucleus that is nicely stained. However, the nucleus appears to be absent in some of these cells. Every live cell has to have a nucleus, but this just points out that these epidermal layer cells are 3D cells, and in some cases the nucleus is located towards the top of the cell, whereas in other cases the nucleus is at the bottom of the cell. And of course, when you use a microscope to visualize these cells, you're only looking at one plane of focus. So you have to move up and down through the cell to be able to see the entire contents of the cell. What we'll need to do now is zoom in even further. So I'm going to use the blue banded objective lens, which is the 40x lens. And this will allow us to look right up at the edges of the cells. Let me talk you through what we can see here. So we can see parts of 9 cells. And if you focus now on the cell wall, so for instance in here you will notice that there is a line in between the 2 cell walls which has not stained very well, and that is the middle lamellae. The middle lamellae is rich in pectin. You might think that there isn't any nuclei visible in this particular photo, but if you look down here, you see those 2 unstained dots. They are the nucleoli of the nucleus. So once you have spotted the nucleoli, then you can quite clearly see the nucleus. The next thing I want you to notice is up here at the edge of the cell. These cells are fully turgid, meaning that the vacuole is full of water. And you can see signs of the edge of the vacuole just here. When you look closely, you can usually see it in almost all of the cells, especially at the ends where cells meet. Notice that the cytoplasm can be easily seen in here in the edge of the cell where it is in between the vacuole and the cell wall. You can also see the cytoplasm down here around the nucleus. The cytoplasm is found throughout the cell, but remember that vacuole tends to push the cytoplasm right up towards the side of the cell. Finally, it's not visible here in this section. However, every student should know where the cell membrane is. And the cell membrane is just underneath the cell wall, and all of the entire cell contents is inside the cell membrane, including all of the cytoplasm and all of the vacuole. So if I was to point out where you might see the cell membrane here, if you were using an electron microscope, it would be right here, right up towards the edge of the cell wall, and of course spreading all around the internal surface of the cell. The next few images will show the journey across the slide, and I'll point out some interesting structures to you as we make our way through the slide. Notice how all the cells are not regularly shaped, they're all different. Notice how the vacuole doesn't reach into the very corners of the cell, and instead what's in the corners of the cell are strands of cytoplasm. Sometimes the nucleus is stained, but sometimes the nucleus is not stained. But now that we're visualizing these cells at 400x, you can see how the cell walls have stained pink with toluene blue, meaning that they are showing a positive result for cellulose. When you watch this video and you'll need to complete some tasks in your workbook, you'll be able to pause this video to get the details that you need for your drawings. Here we can see an almost bubble effect. And what you're seeing there are strands of cytoplasm that are sitting in between the cell wall of the cell and the vacuole. So we are in focus just under the cell wall of this cell. So you can see the strands of cytoplasm spread across the vacuole. Sometimes it's hard to appreciate that these cells are actually 3D cells, but as I move the focus up and down, you will see the cell walls look like they're slightly changing shape, and that's just giving a hint to the overall shape of the cell. We will colloquially talk about these epidermal cells as being a sheet of cells 1 cell thick, but those cells are not flat, and we have to remember that they are 3D cells. And when you have a better appreciation of their 3D nature, it's much easier to picture how there is a large water-rich vacuole inside in these cells, and it is indeed that vacuole which helps to give the cell its shape by pushing against the cell wall. We can go ahead now and look at our epidermal squares that have been stained with iodine. This is what that section looks like when it's magnified at 100x. The brick wall effect is apparent just like in the last sample. What we'll do next is zoom in on 1 or 2 of the cells, and the next pictures you see are the cells magnified at 400x. Here's a single epidermal cell, and I'm toggling the fine focus wheel there to try to show you the 3D nature of these cells again. These cells are turgid, meaning the vacuole is full of water and is pushing against the cell wall, and so there isn't that much detail to see in these cells. For this particular sample, we can see that there is very little starch in these cells. If starch was present, there would be a deep black or brown colour, but the colour of the iodine stain has stayed pretty much the very same colour as it was when it was in the bottle. This is what the cells look like when they've been in 1 molar sucrose for 30 minutes. That bubble area in the middle of the cell, that is the vacuole which has been partially emptied. When these cells were put into a high concentration solution of sucrose, it caused a concentration gradient, and so osmosis occurred where water moved out of the vacuole across the vacuole membrane, which is called the tonoplast, and then out of the cell across the cell membrane through aquaporins, which are channels embedded in the membrane. Water can diffuse across the cell membrane. It's almost like it wiggles through the phospholipids that make up the membrane. But they also have these aquaporins embedded in them. So if we zoom in here at our tonoplast and at our cell membrane, you'll have to use your imagination to picture millions of these aquaporins embedded in those membranes. and they allow just water across, and they won't let any charged molecules across that, like our solutes. You can very clearly see all around the vacuole. And you can see that the cytoplasm has collected around the vacuole here as well. It's important to point out here that this area in here is actually airspace. So there's nothing in there at the moment. And our cell membrane is actually located around here. When you zoom in even closer, you can see that there are contents within the cytoplasm, and they can be anything from vesicles to fat droplets to mitochondria and other organelles which are found in the cytoplasm. They appear to be fizzing, probably not because they're physically moving, but rather because the photons of light from the microscope are colliding with them. That motion is known as Brownian motion. And as I zoom in on this area of the cytoplasm, you can see it a little bit more clearly. That movement could be down to Brownian motion. However, from lectures you will have learned that the cytoskeleton of the cell is responsible for moving organelles around the cell. Let's look at a photo of a cell that has been plasmolyzed using this 1 molar sucrose solution, so we can see indications of where the cytoskeleton might be connected to the cell wall. This is a collection of cells that have been plasmolyzed by placing them into that 1 molar sucrose solution. I'm just going to talk you through the anatomy of one of these particular cells so that you can understand my explanations. This black line here, this is pointing out the cell wall of the cell that I'm going to explain. It's quite clearly plasmolyzed and the vacuole is empty. Here we have the cell membrane. Because the vacuole is empty, the cell membrane has retracted away from the cell wall because that vacuole is not pushing the cell membrane against the cell wall anymore. All these areas are all air spaces. So there's physical airspace in between the side of the cell membrane and the cell walls. Now that we have a good understanding of the anatomy of this particular cell, it is probably a lot easier to appreciate where the most firm connections are between the cell cytoskeleton and the cell wall. They are probably in the areas highlighted here. Of course, there's no way to tell for sure. We know that those other areas are air spaces, and we know that this is where the cell membrane is. So it is probably these points where the cell cytoskeleton is tightly connected into the cell wall. The microtubules and the microfibrils of the cytoskeleton are too small to visualize using our light microscopes. If you wanted to visualize these structures, you would have to use an electron microscope. Finally, what I'm going to do in this practical is induce deplasmolysis in these cells. So as we were just exploring, this is what epidermal cells look like when they have been bathed in 1 molar sucrose solution. So at the moment they're plasmolyzed. Their vacuoles are empty of water, and this might be what a droughted plant looks like. So if you were to look at the leaves of a droughted plant, you might see signs of plasmolysis in those cells. To add the water back to these cells, all I needed to do was drop a few drops of fresh water on the side of the cover slip, and then all you need to do is to hold a tissue on the other side of the cover slip, and that draws the water in under the cover slip and around your epidermal section. This is the point where I added water to the cell. These cells now find themselves in a situation which is quite the opposite of where they find themselves with the 1 molar sucrose solution. Now water has to move back into the area of high concentration, which is now the inside of the cell. And so water passively diffuses across the membrane, across the tonoplast and into the vacuole. I've sped up the video at this point because it did take 10 minutes for this to happen. So what you can see here is an accelerated view of what is happening during deplasmolysis. This is what would happen if you were to water a plant that wilted. It would take up water through its roots, it would move up through the stems, through the petioles and out into the veins and the leaves, and eventually it would reach into the cells and start filling the vacuoles. Thanks for watching the video. I hope what you learned in the video helps to put in context what you hear about in class. Best of luck in your practical submissions. Make a really good effort. I look forward to seeing what you produce.